Good day, teacher Sandra and classmates. Our group is made up of Diana Sofía Roa Rodriguez and Daniela María Chaparro Carreño, and today we will expose to you about genetic engineering.
Genetic energy is a thinking in which is used recombinant DNA technology composed by fragments from different sources that have been mixed in a laboratory. To introduce new traits into an organism, this is about in the genetic codes that all organisms share in common, expressed in the four nucleus building blocks involving the gene encoding of proteins, whether it is made in an animal, a plant, or microbe. One of the first causes occurs in the 70s where it marks the star event called Stanley Hawking cloning technique. Thanks to this, he began to explore the field of bacterium plasmid. He location to understand how the genes included in the plasmid make the antibiotic-resistant carrier bacteriums. All test results focus on the development of methods to combination and transplant genes. This cause of the research to implement development and innovation in their some technique to produce different results.
Genetic breeding methods. GE or genetic engineering is used only when all other techniques of conventional breeding have been exhausted, as cases that the trait required to be introduced is not present in the germplasm of the crop. In this way, the genetic engineering applied to crops breeding is commonly known as transgenic or genetically modified GM crops. It is based in 5 main steps. First, extraction of DNA from the organism that present the trait of interest. Second, gene cloning to isolate the gene of interest from the DNA. Third, design and package of gene to control and properly express the trait in host plant to produce modified gene in mass into a host cell. Fourth, gene package is introduced on cell plants by different transformation processes, as biolystic transformation using a gene gun or through agrobacterium-mediated transformation. Fifth, known as backcross breeding, is the final step where transgenic crop is crossed with an important agronomic traits variety and selected to obtain high-quality plants. The use in agricultural systems. Basic technologies of genetic engineering in plants are related with molecular biology, bioinformatics, genetic transformation, and vegetal tissue culture. As example, the author Chaparro Giraldo in 2014 relate mechanisms to breeding rice crops applied in Colombia, where first it is obtained the CRY1AC gene sequence into a web page. It is a gene that gives resistance to the plant against Lepidopterous insects. Then it is necessary to design a new sequence more efficient to rice crop, including the expression cassette generation that contains promoter and finishing region. This cassette is sent by email to a foreign company that synthesizes the construct, introduce it into a pre-selected transformation vector, and send back it to the laboratory in Colombia, where has been developed the GM variety of rice. When it is realized the gene transference through biolystic transformation or agrobacterium-mediated transformation, where is obtained transgenic cells that allow regenerate a complete plant. by techniques of vegetal tissue culture. Methods for transformation. The biologistic methods is a method that allows direct delivery of foreign DNA, RNA, or protein into plant cells, as authors Mousavi and Ambraje-Ford in 2019 explain. In this method, gold or tungsten fulfill the function of receive the precipitated DNA and transfer it directly into the plant cells and tissues. In this way, it is possible to introduce new traits to plant species with a high degree of heterozygosity, as some fruit trees. One example of biologistic transformation in fruit plants was realized in pineapple. The first experiment that authors explain is about introduce genes for herbicide resistance, in which they introduced a system for transformation of protocorm-like bodies with Gus and PT second genes, confirming the gene insertions at 1 to 3 loci by southern hybridization. In the published results, the scientists proved that this pineapple CV pocket to an herbicide with glufosinate ammonium as active component can be resisted by transforming with BART gene using biologistic transformation, and its stable integration into the genome was confirmed with PCR, RT-PCR, and southern analysis after 318 days. Via agrobacterium-mediated methods. It is considered that plant transformation by agrobacterium tumefacens is better than biologistic one due to a higher frequency of intact single-copy DNA inserts. In the research, Richardson T. et al. In 2014 proved to use agrobacterium transformation to produce selectable marker-free transgenic plants in elite germ plants by using no selectable marker and no selection throughout the transformation process. They plant wheat cultivars under glasshouse conditions and were collected the wheat heads at anthesis and harvested 12 to 14 days post-anthesis for transformation. At the same time, binary vectors were introduced into agrobacterium strains AGL1 or GV3101 by triparental mating. Also was used E. coli helper strain RK2103 and E. coli DH5 alpha that contains the binary clone of interest. All were mixed and cultivated into an agar plate of LB culture supplemented with antibiotics. For transformation, seeds by each cultivar were harvested and surface sterilized. The embryos were removed from seeds and then were co-cultivated with agrobacterium strains that contain binary constructs of interest per 2 days on WLS AS medium in dark. After that, embryonic eggs were transferred to an WLS P5 callus induction media, and 5 weeks later, they were transferred for another medium to root development. As summary, they concluded that the ability to introduce transgenes into cultivars as wheat without a selectable marker potentially enables the rapid incorporation of transgenic traits into breeding programs. Finally, to consider an organism as transgenic, it is necessary to check that the inserted gene is stable, inherited, and expressed in subsequent generations.
The use in a livestock system. Genetic energy has characteristics that are included in livestock farming, where there are plants of genetic improved between team. Better called conceptual use genetic energies along with conventional breaking science their methods facilitation genetic improvement for the instinct to create enough birds of animals that again more weight with less food, faster growth, with greater production of milk, of meats. With less fat, the biggest controversy comes from the cloning of the famous doll shape in 1997. In this diagram, we observe, for example, first cloning of the genes, second insertion of recombinant DNA and embryo implantation, third transgenic cow with the UFAX8 genes, fourth ophthalmic milk factor 8. For example, in this second diagram, we observe four characterizations and transgenic is injected into the nucleus of a zygote. Second, embryo is implanted. Third, only some goes with carried the transgene. Fourth, milk is extracted with the desired protein. These diagrams and the one and second so breed of animal that's more with the goat production the use system.